And absolutely delighted to be here with all of you today, having the opportunity to talk to you about some of my current research in collaboration with the Center at Virginia Commonwealth University, the Center for Clinical and Translational Research, a group of psychiatrists, neuroscientists, and computer scientists, and statisticians working on what it is really one of the probably most relevant, most significant societal problems, which is drug addiction. So we are working with large amounts of data, imaging, trying to convert the data into valuable knowledge that can help us to address this issue. So let me start talking a little bit about addiction. So addiction manifests when we are craving for an object, the object of addiction, and we start losing control over its use, and then we start losing also judgment, and we make decisions that we know would be not healthy for us and would lead to non-desirable outcomes. So what happens with addiction is that it does change our brain. It's really a brain disease. So when we think about cardiovascular problems, we all acknowledge those are diseases of the heart. If we are thinking about diabetes, we acknowledge those are diseases of the pancreas. It's no different with addiction. Addiction changes the metabolism in the brain. The machinery in the brain is altered, and we need to help those individuals to get that back to normal. So it is a disease of the brain. It's still by many considered as a choice. So there are choices that will lead to certain diseases, and that's the case here. It's no different than potentially making poor choices about exercise or diets that could lead to a disease. So it's the same thing with addiction and punishment. Punishment or the expectation of development of willpower would not take us there. We need to have a better understanding of how the brain has been altered and then develop proper treatments that can help us to take back to normal. So in this case, we are focusing on cocaine. So cocaine can be derived from coca. That's a plant that we have in South America, primarily in Colombia, or it also can be prepared synthetically. It's an illegal stimulant, and it can be inhaled or injected or snorted. Just few statistics for you. This is in the United States, but the number of overdoses has been rising 5.5 every year since 1999. We have every day 100 people dying from drug overdoses, and this is a problem that is impacting close to 22 million individuals in the United States, and is 1 in 5 the number of young adults who are users of illicit drugs. So this is a problem that right now is non-stoppable and continues to grow. So let me explain a little bit how it works. What happens in the brain is we have in this middle section of the brain, in the limbic system, we have what is called maybe the pleasure or the reward system there. So we have what is called the nucleus accumbens, and in that area, there is a substance that is being emitted once we are exposed to some type of rewards. Sex, food, money. So that would release dopamine. That's the name of this chemical that is being released when we are being exposed to these natural rewards. The same thing when we're exposed to cocaine, but the levels of dopamine could be potentially 10 times greater than with sex or food or with money. So it does lead to great pleasure immediately. So these high levels of dopamine, they are here in this reward system in the brain, but next to the nucleus accumbens where we release the dopamine, we have the hippocampus, and I see right here, and the amygdala. The hippocampus controls memory. So what happens is that the memory of the pleasure that is linked to these natural rewards or the cocaine is stored in the brain. And when we have these very high levels of dopamine, they are floating in all this system, and they alter that. So the hippocampus keeps bringing back these memories of the pleasure. Next to it, we have the amygdala that controls anxiety and actions and potentially judgment. So by having these high levels of dopamine when we're exposed to cocaine, this system in the brain starts getting to be altered. The machinery of the brain is not functioning properly. So again, the hippocampus, anytime that we have exposure to some stimulants, an environmental stimuli, is going to bring back those memories of pleasure, which starts again altering, losing control, and then the judgment. Also, what happens is that if this is not changed, if this doesn't go back to normal, we are in the same place. And that's why 60% of the people that go through the recovery, they relapse, they go back to it because the brain is still altered. So the goal of this study is to get to understand better the brain, to look closely at the differences between the brain of an addict versus non-addict, and that will help us to start developing proper treatments that can help those individuals. So we do have access to fMRI, so different type of imaging. So the fMRI is functional magnetic resonance imaging, and then we also have access to the type of new imaging, DTI, diffusion tensor imaging. So in our center, we have been able to continue using with the working with the latest developments that we have in imaging. And we work with different ones. In this case, I'm going to focus on DTI. The fMRI gives us the information about the blood flow in the brain, the gray matter. So it's very helpful to understand how we respond to different stimuli. The DTI gives us the white matter, so that helps to understand how the nerve impulses are transmitted in the brain. So it helps us to understand the activity, the machinery of the brain, the structure of the brain, and that way we, in this case, we are in a position to better understand differences in the brain for addicts versus not addicts. What I also want to mention that while in this study today, I'm going to focus on imaging and how we are using that to help us to understand better the brain and develop with the psychiatrist proper treatments for these individuals. On parallel, what I've been doing and also working with the General Attorney Office in Virginia to be able to help these individuals, drug addicts, and to use education, to use literature, to use writing as a way of rehabilitation for them to find other different type of rewards that can help them to lead to recovery, self-reflection. So we get 10 drug addicts every year that instead of serving their time in jail, they come to us, to the university, and we help them using education as a powerful tool for rehabilitation. And, with a lot of lawyers and a lot of control, too, as we go through the process. So in the context of the imaging, so we have these images, and there are really two ways that maybe we can try to develop here. A statistical framework to understand the differences in the brain of an addict versus not addict. So we could use this image that is large amounts of data. It's going to be 3D, and we can use that to study some clinical outcomes, or we can use the image on its own as a response and try to understand how, in this case, we're interested in addict versus no addict, to be able to make inference about the impact of certain covariates or predictors on the image. And that's the later. So here we're going to be working with the image on its own as a response with the purpose of making inference about the differences between being an addict or not addict, what areas of the brain are significantly different between these two populations. So these DTI images are 3D. So here you have the projections of how it would look like across the different dimensions. And so we have, in our study, we have a group of addicts and no addicts. Here we are working again with cocaine, 58 in total. So we work with these individuals prior to even putting them through the imaging. We conduct psychological studies to properly classify them. So we go through a very thorough process to make sure that we classify properly individuals into addict or not addicts. We really want to make sure that in making inference we have good classification, and we go also through some clinical studies, urine samples. So this is not just self-reported data. But we also have information about gender, age, education, the cocaine use, and ethnicity. And again, the primary goal here of these studies is to be able to make inference about the drug use on the brain activity, and the follow-up is working again with the psychiatrists on the development of the proper treatment. So what you see here are slices of the brain along the different dimensions. In the top row, we have it for the non-addicts, and then the bottom one for the addicts. So here, just maybe to think, I don't think that the laser, you can see very well the laser, but just to point out to you where the dopamine gets released. So it will be in this area right there is where the dopamine will be released. So again, these are slices of the brain. The issue is that what do we do with the images, right? Because this is a lot of data. Each one of these images will be about 8 million voxels. A voxel is sort of like a pixel, a unit of information. So it's very high dimensional. And if the purpose is to really understand the brain, the spatial structure in 3D of the brain, we certainly don't want just to project that into one dimension and start working with a vector or even with a matrix. So this is really the main challenge, how we can preserve the spatial structure of this image to be able to make inference about the differences between the two populations, but in a way that we can manage the large amount of data that we have here. So for the most part, here parametric approaches would then be feasible when we have about 8 voxels for each one of the individuals. We could, a lot of the work has been done within neuroimaging has been really focused on maybe in just a particular part of the brain, or aggregating data, or creating a vector, or projecting into a matrix. Those are the type of approaches that we commonly tend to see. Here we are going to try, the focus is to preserve that structure, because there's still the reality is that we don't know that much about the brain, so we are not in a position to determine, well, we are going to aggregate here, or this is the area that we want to focus. We really want to focus on the entire brain, because part of what is happening is there are certain parts of the brain that are impacted that we are not aware, and that's the focus of the study. So we also want to take into consideration different sources of uncertainty about the data and the models that we're going to be working with. So with all that in mind, we want to introduce a Bayesian framework preserving the structure of the data. So this is 3D, a 3D array or a tensor, and we want to work with a tensor. So this is going to be, to my knowledge, the first Bayesian tensor regression setting using DTI as the response. In the context of the literature, there is some literature working with these 3D arrays. For the most part, it's not having the tensor of the array, the image, as the response, but as the predictor. And in the context of having it as response, haven't seen anything in the Bayesian literature, but what I've seen is having the tensor as response, using a frequencies approach in those type of settings to be able to implement it. You need to have a large N in relation to the dimensionality. It's certainly not the case here. These type of studies with drug addicts are incredibly expensive and difficult to implement, and the fact that we have about 60 is certainly quite an accomplishment, but that in relation to the dimensionality that we have here is still very small and in relation to P. So these type of frequencies approaches would then work in this type of setting. So that's why we are moving forward with this Bayesian framework for a tensor response, and we're going to have to introduce certainly approaches that are going to help us with dimension reduction. Okay, so the first thing I would do is to offer a little bit of background about tensors and how to work within. So we have this 3D image. It's a tensor in 3 dimensions. And then we have the units. The corresponding units are called voxels. And you have here a voxel here. So they have 3 coordinates in this setting. So once we have this image, or it could be in M dimensions here, we are working with 3 dimensions, but it could be m dimensions. So we can unfold the image into a matrix. We can always do this unfolding. So that's called matricization, and it can be done in different ways. You can work with horizontal slides or lateral frontal slides. So you are going to have different type of matrices depending how you do the unfolding. So when you have tensor of order 3, there are 3 ways to unfold the tensor. The matricization could be done 3 different ways. That is called mode 1, mode 2, or mode 3. So the mode 1 at the top would be when we are working with top slides. So if you look at the top slide in that cube, the top slide will become the first row. So it's just working with horizontal slides. That's the way you do the unfolding. If you work here with the lateral slides, so if you look in that cube, what would be the lateral slide? That would be become the first row in the matrix, so that becomes the mode 2 matricization. Then you can work with the frontal slides. If you look at what happens in that cube, the frontal numbers, that becomes the first row, so that will be the mode 3 matricization. So there are different ways that you can unfold a vector, tensor to create a matrix. Another important concept that I need to introduce is how you do the product that we're going to be doing in regression of a tensor with a matrix. So that's called the Keynote product. So when you have a tensor of, in this case, order 3, but really of any dimension, you can always extract vectors. Vectors are called fibers. Vectors are tensors of order 1, and we call them fibers. So we can always, from the tensor, extract fibers. So if the product that we're interested is of K, this order K product. So what we do from the tensor, from A, we construct those fibers of vectors such that we keep all the dimensions but the K dimensions. So we vary the K dimensions. So we create all those fibers that we call of mode K, and we multiply each one of those vectors by the matrix. That's how we do this multiplication. We create the fibers, or the vectors, and we multiply each one of them by the matrix. Use the matrix multiplication. To be able to do the multiplication, you need to make sure that the length of the fibers is the same as the number of columns that you have in the matrix. And that's how you do the multiplication. So let me show you with an image an illustration of how that works. That's very relevant here because we are going to be using that quite a bit in our tensor regression. So we have we have the tensor here in 3 dimensions, and what we do is we extract the fibers of mode 3 because that's the product that we want of order here. This is the product that we want. So we want to keep all the dimensions fixed by the third. So here is dimension 1, 2, and 3. So we are changing this dimension, okay, up and down. So these are the vectors that we create. These are the mode 3 fibers that we have, where we keep everything fixed, but the third dimension up and down. So we create those fibers, those vectors, and then we multiply each one of them by the matrix using matrix multiplication, and then this is what you get. Okay, so when you multiply each one of those vectors by the matrix, you get this tensor. So this is tensor multiplication. So again, the tensor multiplication can be done of different modes. So this is the mode third, with and again, that's by creating the mode K fibers, the vectors where you keep everything fixed but the K dimension. Okay, so with that in mind, we can move into the tensor regression setup. So here, I'm going to introduce this in general terms, and then we'll go back to our image where we are in 3 dimensions there. But in general, you could have m dimensions. So you have a tensor, a response for each one of the individuals. So the index for the individuals would be I. So we have n individuals, and for each one of them, we have a tensor response, y. Okay, so this is our tensor response. So here, the type of inference that the focus again in this problem is inference. So what we are trying to do is to be able to understand differences, again, between addicts and no addicts, and the other variables of interest, gender, ethnicity, and other different things that we are also interested. So those are our predictors. And then we have residual term that we want to put some potential dependence that may be left, residual dependence into the error term. But here are the betas that are the focus, again, inference. We want those betas to change across the brain. We want to understand, again, what parts of the brain are different for the addicts versus no addicts. So the betas need to change across the brain. The betas need to change also across the predictors if we are trying to make inference about different populations. So as a result, then if the betas are going to be changing, then they are going to become a tensor themselves, and actually of higher order because they are not only changing across the brain, but across predictors. So if the response is of order m, a tensor of order m, the betas are going to be of order m plus 1. Okay, so those would be our betas. Difficulties in the laser here. So the betas would be at m plus 1 tensor, and we wanted to change, again, across the brain and across the predictors. But we also want to characterize the dependence that is left in the residual term. So we want to introduce some covariance structure here in, let me see if I can get a better laser. So here we also want for the residual covariance structure, but again, this will be very computationally expensive in the m dimensions, assuming here separability. Okay, so let me explain you what we are going to try to do, since we need to reduce a little bit the dimensionality that we have here. So I'm going to explain the dimension reduction tool that we are going to use just in one dimension, and then we'll take it to our setting. So suppose that our response is just a vector, so we are in one dimension, and this is the response that we have for each one of the individuals here. So what happens is that in the response, again, one dimension here, in the response space, there are some parts of the response that are relevant here in these regression problems, and there are others that are not. They're not so relevant because there is potentially a part of the brain that is not going to be impacted or affected by addiction, right? So here they're saying, so there is some components here, some linear combinations in the response space that are not going to be dependent or sensitive to changes of the predictors. And there are others that are very much dependent on the predictors. So we want to focus on what is relevant for the prediction here, the part that is material to the regression, the linear combinations in the response space that they are sensitive to changes of the predictor. So that's what we want to focus here. We want to maximize our focus on this material part to the regression, what is relevant. So here would be, as you can see in this setting, there is part of the response, the yi, that is not changing with the predictor. Okay. So what we would like to do is to be able to project ourselves into that space that is material for the regression, or that is relevant for the regression, and we want to work there with the hope that as we do that projection, as we go into this space, we go into a much lower dimensional space. So, so we have a subspace of interest. Oh, that's where we want to be. That's the part that will be relevant for the regression setting, and we have a projection that is going to take us there. So P of O will be the projection matrix that is going to take us where we want to be. On that space, that depends on the on X, that depends on the predictors. This symbol indicates a statistical independence, and this one is statistically identically distributed. So there are going to be some basis functions that are going to take us in that space that is material for the regression. So the gammas will be those bases, and you can see that we go, if the original dimension, in one dimension in this case, was R, so R will be millions or very large number, then these basis functions will take us into a lower dimensional space U. So U is expected to be much smaller than R. So then if we look at the covariance function that we have here, so the covariance function could also be written as a function of this part that is relevant for the regression, the variability that is material to the regression, the variability that is immaterial or irrelevant in the context of the regression. So this is the setting that we want to project ourselves into a space where that is relevant for the regression that we're doing. Okay, so what happens is that then with these basis functions, the gammas, that they are the ones taking us into a lower dimensional space, the betas, or the coefficients of interest, again, the purpose is inference, also are projecting to this lower dimensional space. So here the betas are projected into the lower dimensional space. Remember that the basis functions were r times u. U is the lower dimensionality. So here, really, we are going to be working with these etas, which will be the coefficients in this lower dimensional space. So a few things to keep in mind here. The span of the betas, which is the different type of linear combinations that you can create in the vector space, will be included in the span of these basis functions. And then we have conditional independence between the part that is material and the part that is immaterial for the regression. Okay, so now we can take this into a higher dimensional space. So this is the basic setting in one dimension, which is, again, is this reducing the dimensional, introducing this sparsity principle to be able to allow us to work with this large amount of data. So in the context of why this will be applicable here, it is expected that when we are working with any type of brain disease, not just addiction, but there are parts of the brain that may not be impacted by addiction. So here we just want to focus on the parts that will be relevant here. Okay, so I have introduced this in one dimension. We need to take it to M. So we have a series of subspaces. So we have been doing this now, and when the response is 1D. So now we have M and M, mod M tensor response. So we do the same thing for each one of the dimensions. We find that different projection that will take us into the lower dimensional space. So that's what you see here. Across each one of the M dimensions, we go through the same exercise, and we find the projection matrix for each one of them. And then we put it all together. So that's what we have here. For all the different dimensions, we bring it all together. And that would give us, when we look at it all in a comprehensive manner, we have all this material part for the regression of the response space. So a way to maybe represent this is the way I have it here, where you have each one of these basis functions that, you can think about this as a high order PCA, where you have the components. It's what each one of the components, right? So these this would be each one of the eigenvectors that you have, or the principal components that you have in a PCA approach in high dimensions. And this would be what we call the core tensor of the projection of the tensor in the lower dimensional space, that in a sense facilitates the explanation of what type of relationships you have across all these different principal components. So that's a way to think about it. So here, maybe another way to maybe simplify a little bit or illustrate what we're trying to do. Again, this is a high order PCA, tackle the composition, a low rank the composition. So so at the end, what we have is our tensor A. What we have is this projection of the tensor into into a lower dimensional space. And then the different components here. Okay. So let me see if with this graph I can illustrate a little bit better this decomposition. So here, this would be the tensor response in 3 dimensions, and then we have the principal components, so these basis projections here, that would take us, each one of them, from the unal dimension R into U. And then we have this core tensor that will be the projection of the unal tensor into this lower dimensional space. So again, what is key here is that U will be smaller than R, okay? Okay, and then with the covariance will be the same thing. So the covariance can be represented as a function of this material part of the regression, as we have seen in one dimension, and the material part of the regression. So we can do that for each one of the covariance components here. I'm sorry. Just in the fly. You have 25% chances that you get sick when you're flying, so. So here, let me spend a little bit more time with the covariance here. So we have these covariance in each one of the dimensions. Again, we are projecting this into a lower dimensional space. So a way to write it down is in terms of the basis functions, the gammas, and then this fully symmetric matrix here in the lower dimensional space. So this is in the U, in the projected space. So the gammas could be fully symmetric, and these, the omegas could be fully symmetric, and then the gammas would be semi-orthogonal. Okay, so some of the priors here for the for the coefficients of interest, the etas in the projected space. So we are going to work with tensor normals, which is really a vectorized normal prior distribution for the coefficients. The reason why I want to talk about the priors briefly is because we are able to get conjugacy, which is, as you know, critical in this setting to be able to implement it. And then for the matrices, we are going to work with this type of priors, matrix Bigman, that would give us, again, as I said, the conjugacy, so we can implement Gibbs sampling. What is of interest here, too, is the dimensionality of these envelopes. They use the dimensionality when we project ourselves into this other space of lower dimensionality. So what we are doing here is we can use different type of cross-validation information criteria to be able to determine what is an appropriate value of those the use of the dimensionality. Here we're working with DIC. What I do want to point out that the focus again here is inference, and we are using this as a tool for computational efficiency. So it's not critical for us necessarily to optimize this to determine what is the ideal size of the envelope, as long as we are able to get some reduction in dimensionality and being able to implement it. So I'll show you some results with regard to how well we do with DIC to be able to get a proper dimensionality, but the results are not really sensitive to that. What it would be sensitive to that, it would be the computational effort. If we are in a position where we can go from 200 to 20, in the reduction of the dimensionality, and instead of that we go from 200 to 100, we are not going to benefit so much computationally. I mean, at the end of the day, we'll still be able to make inference, and that would be impacted, but the impact of not being able to go to the lower dimensional space is primarily in the computational effort. Okay, another contribution here too of our work is making sure that we look carefully at statistical properties. So here we want to make sure that we have consistency of the posterior. So we start with some imprecise, potentially, information about the parameters with our prior distribution, and we want to make sure that as we gain knowledge with the data, that the updated knowledge as characterized with the posterior becomes more and more accurate. So that's the concept of consistency. And here what we'd like to be able to do is to show that the posterior, as we increase the amount of data, so here the posterior of the that we have here in a neighborhood, in a covariable neighborhood around the truth, that the procedure is going to take us there with probability 1, right? So that's the concept of the consistency. We want to make sure that the posterior is going to be there in that little neighborhood around the truth with probability 1. And in this setting, we are able we're able to prove that instead of working with the neighborhood, we work with the complement of the neighborhood. So what we want to see here is that this is the complement of the neighborhood. So we want to see is that we are never really going to be there. We're going to be there with probability 0. That after increasing the amount of information, updating our knowledge, we are going to be able to get to the truth. So we'll be in this complement of the neighborhood where the truth is with probability 0. So we're able to derive that, which again is relevant in this type of setting with large amount of data to make sure that we can rely on the type of inference that we're making in the posteriors. So let me show you a simulation example to get a better understanding of what we're doing here, and then we'll move to the data. So again, this is Bayesian tensor regression. So we have our response of tensor of order 3 in this setting, and then we have the betas. The betas will be a tensor of order 4 with different predictors and their residual component, the error term. So here we have, in this setting, we want to see what happens when we have different type of envelopes, so the dimensions in the projection, the lower dimensional space. So these will be the different true envelopes that we're working with. Originally, we have 10 by 10 by 10, and we have a sample size of 100 and 400. So we are going to compare here different settings. So we have the Bayesian regression setting with our response here again, the betas, tensor of order 4. And we are going to implement just a Bayesian OLS approach, in which we assume independence in the residual component. So we neglect here to characterize potential dependence that was left in the residual term. Then we are going to use wavelet approach, which is with 8 by 8 by 8, and to characterize that dependence in the residual term. So we are building, right? So we start with a Bayesian OLS, then we add a covariance structure to it through wavelets, and then we are going to compare that with the method that we propose here. So the first thing is to look into the computational effort to see how much we lose once we go into this approach where we characterize the dependence, the covariance that at the end of the day, that's really what we are doing here with our approach. So if we compare the Bayesian OLS versus the wavelet versus the proposed approach for the different settings, and here will be the number of observations, and this is the true size of the envelope, we see in seconds how long it would take to run this approach. And we are losing here in the context with regard to the limited characterization of the structure with the wavelets is not very different than we're using wavelets in this setting. If we start looking at what is of interest here, which is the inference that we are making, so we are looking at the beta. So this is the average and the standard error of the error, the deviation of the betas with regard to the truth. That's what we have here. So we have mean standard errors and the coverage. So in blue, the lower row here would be the coverage in each one of the settings. So we are doing the same thing. We are comparing the Bayesian OLS with a wavelet with a proposed approach for the different type of envelope sizes, the different value of n. So these will be the results for this error in the estimation of the betas, and then again the standard error and the coverage. So what I want to point out, so there is clearly a reduction as we want. We want these to be as small as possible, right? The difference between the truth and the actual estimated beta to be as small as possible, and there is a remarkable reduction pretty much across the board with the proposed method to the point that it will be 50 times smaller. But what I want to point out is the coverage that we have, that with the wavelet, in some cases the coverage is 50%. The nominal coverage here was 90. 90%. This is just looking at the MSE. This is in-sample prediction error. So this is in-sample. We'll look at out-of-sample next. So very similar results, very remarkable reduction here in the within-sample prediction error here to point them, in some settings, particularly when we go to the lower envelope sizes, it could be of 10 times reduction. Not so much when we are here in the settings that are more similar to the size of the wavelets. And then out-of-sample prediction error here. So here what we see is, again, the same thing, the error in the out-of-sample prediction and the standard error. And in this type of setting with smaller envelopes is where we also see a more remarkable reduction here in the prediction error. It becomes a little more similar once we go to the larger envelope sizes that correspond to the size of the wavelets here. So let's talk a little bit about the reduction in the dimensionality and how to find an appropriate value of, in this case, we are calling it U. We go from R to U. So again, the purpose here is not to find what is the best size of the envelope, but we really want to be able to optimize in a sense that we get as close as possible to the lowest size. So here what you see is just a simulation to see what we are doing, if this is helping us to get where we need to be. And what I'm showing you here is that for this is again a tensor of order 3, and the actual envelope size was 2 by 2 by 2 across the different dimensions. We started with an order of size 5 by 5 by 5. So what you see here is the number of times that you choose 2 as the size of the envelopes. So here are the different sizes that you could choose. So you can see that for the most part you tend to choose 2, and if not very near 2, and you don't seem to ever go into the extreme. So it seems that we are capable to identify the dimensionality, or being in the neighborhood of the dimensionality of interest. We have been also looking at sensitivity to the priors. This is in the same setting as the DIC that I just presented. And it seems, so here we've been changing a little bit the covariance, the mean, and it seems that the results here in the context of looking at the error in predicting the betas, so the error in the prediction, that it seems that the results don't seem to be very sensitive to how we initialize the priors for the betas. Okay, so let's move now to the application. So again, here what we are doing is we are looking at the DTI, so these very large images. So we have 8 million boxes for each one of these individuals. We have 58 individuals, a group of addicts and no addicts. And what we are trying to do is to be able to identify differences in the brain for these two different populations. Okay. So we implement our proposed Bayesian tensor approach with the dimension reduction, and the DIC approach here identifies going from, in each one of the dimensions we have about 200, so here we're going from 200 to 20 in each one of the dimensions, and which is a remarkable reduction as we expected. And we are looking here at the within sample prediction error and out of sample. If we compare the Bayesian OLS with regard to the proposed method, you can see the remarkable reduction here in the error, which points to the fact that we certainly benefit by having these residual dependence in the model. So now what becomes really challenging in working with these images and now start looking at the results and being able to start identifying what areas of the brain matter or not, how we look at all these coefficients in a very these large images. So what I'm showing you here, it would be what is of interest, right, to look at the differences in the brain for the cocaine addicts versus not addicts, what areas are relevant or not. So if you plot these are slices of the brain, and if you plot, what would how it would look like here for the what areas would be different for the addict versus not addict. you start and you look at the C-scores, really, in a sense. Here we have the coefficients, the standardized coefficients, the coefficients divided by the standard deviation. And in red, you see the areas that matter. So it becomes really difficult. These are just slices, right? And you start seeing potential dots in different areas. So it becomes challenging to identify what areas really matter, and there is an issue with false discovery rate. So what we do here is we, across all different slices in each one of the 3 dimensions, we look at the 10%, the highest 10% that are relevant. So we look at the we try to focus on those 10%, 10 higher percentiles in the context of the significance, and that's what we are plotting here. So across each one of the dimensions, we are looking, and those will be in red, the areas where we have significance across all the different slices. So we look at, what would be the 10% highest in the context of significance, and then we plot in here to identify where we have significance. And there will be a few dots that you see here in some areas here and there. But that wouldn't be enough if you just have a dot here and there. In the context of the brain, we want to see a little bit more of an accumulation of significance, and at least having 15 in working with the psychiatrists recommend to have at least a cluster of 15 voxels where you have some significance. And then in looking at those sort of subregions, so groups of voxels with 15 points, we can easily then go through the atlas of the brain that this was developed by Johns Hopkins, but then we can clearly identify different areas of the brain looking at the atlas and determine what type of functionality we have there, and then start again the process of identifying treatment. So here we work, what I'm showing here is those groups of at least 15 voxels across each one of the dimensions where we have significance here, and that's what you are seeing here. So we see, so these would be in this area, that again, this is where we have the amygdala and the hippocampus. So here in this area, we see certain functionality. What is new here to see is the bipolar depression, an area that controls bipolar depression, and of course judgment too. And then this is coordination. So we start seeing some areas of the brain that we were not aware that potentially could impact addiction, or that would be the result of addiction. And that's where we are right now, trying to understand how to treat those areas, or why that has been impacted by addiction. What I also want to emphasize is that when we do the Bayesian OLS, and we go through the same process, but with assuming independence there, this is what we have. So we are not able to find those significant areas. We're not be able to, in this setting, to find any significant, those clusters of 15 in this type of setting. So again, just to summarize what we have here. So what we have introduced is a powerful approach to be able to handle data in large dimensions with complex type of dependency structures, and by having this computational efficient approach that should lead to reliable outcomes, again, by looking here also some of the properties as consistency for the posterior. And again, this is primarily inferential tools with the purpose of being able to make inference in these high dimensional settings. But more importantly, and this starts helping us to gain a better understanding of how the brain of addicts is being altered, and with the purpose of being able to introduce the treatments that will help us to address this very important problem. Thank you.